that in my mind are really at the heart of the problem of many neurodegenerative diseases. So the first question is that, I mentioned that a subset of ALS is caused by mutations that are inherited in families, and remarkably these mutations or the genes that are mutated are present in most cells in our body, yet only motor neurons are sensitive and they degenerate in the disease, and we don't really understand why other types of cells are resistant to these mutations. So that's one very important question. And then the second question is that these mutations are also present in the cells throughout the lifetime. Yet these diseases develop only later in life. So you can be born with a mutation that will cause ALS. You will have no idea that your motor system is impaired in any way. You can actually be excellent athlete like Lou Gehrig, and then eventually your system starts to degenerate and motor neurons die for some reason. So we again don't really understand what is contributing to this late onset. And so in order to start understanding what underlies the motor neuron degeneration, I believe that we really need to understand what makes motor neurons different from other cells in our body. And but that is and has been always problematic, because motor neurons are found inside the vertebra, so they are not accessible. You cannot watch them. You cannot make biopsies and remove them because that would be detrimental. And so the only way how people studied this disease for many years is by looking at post-mortem samples in deceased ALS patients. But recently we started realizing that there might be other ways how to access this cell type which is not very accessible. And that's by learning from the normal development that happens in the embryo. So we know that, that all cells in our body originate from a single fertilized egg. This egg has to divide, and somehow during the embryonic development, some cells have to be instructed to become motor neurons. And so just to illustrate you how beautiful this process is, I'll show you this video of developing a frog fertilized egg. You see that the cell starts to cleave. The cells are dividing pretty symmetrically, so they all divide at the same time until they form this ball of filled with many different cells. But then something happens. At this point, these cells still look very similar to each other. But then something happens, and this ball of cells starts to undergo gastrulation. This is a process when different tissues become separated and they acquire distinct identities. And you can actually see that at this point, there is a part of the tissue that folded and formed the initial neural tube that will give rise to the entire central nervous system, with the head developing here and then the spinal cord developing in this region. And so how these cells become instructed to become different parts of the nervous system, or nervous system versus skin cells or blood cells, this has been something that scientists studied for now many decades. And the idea is that if we understand what instructs, what educates cells, what type of cell type they should differentiate in development, we might be able to recapitulate the process and make our own motor neurons. So in order to make our own motor neurons, there are two conditions that we need to fulfill. First, we have to have access to naive cells that are pluripotent, meaning that they can give rise to any cell type in our body. So we have to be able to grow these cells and use them then for the differentiation. And then the second problem is that as these cells divide, we have to be able to identify or know what are the signals that instruct the development and cell identity of these cells as they undergo development. So the first problem has been actually solved now quite a few years ago, where researchers such as Gail Martin and Martin Evans identified conditions under which they can take actually relatively young developing embryos. So they started with mouse embryos 4 to 5 days old. That looks like this. This is basically a little hollow ball with a set of cells inside that will give rise eventually to the whole embryo. And when you take these cells, they found conditions under which you can grow them in a petri dish. these cells divide and they can grow, basically you can grow unlimited amounts of these cells. Now we can even get by without relying on these early blastocysts. Actually, Jamie Thompson found ways how to take blastocysts that are discarded from IVF clinics, from couples that already went through pregnancy and they do not need any more pregnancies. And they took these discarded blastocysts and actually shown that they can derive also human stem cells like this from early blastocysts. But there's always ethical issues with using even discarded blastocysts, and so most recently, actually researchers such as Shinya Yamanaka found ways how to take other cells from adult organisms. So we can take a piece of skin or blood cells and then actually reprogram these cells into cells that have the same properties as embryonic stem cells. And so these embryonic stem cells are amazing because, as I said, you can produce unlimited supply of them. They can grow for years in a cell culture in petri dishes. They are pluripotent, which means that they can give rise to every single cell type in our organism. So they can give rise to every type of neuron, skin, heart, muscle, anything. And finally, because we can grow these cells in cultures, we can also engineer them quite easily. We can add genes into them or remove genes and study what these manipulations, what effect they have on the function of these stem cells. So now we have cells that from which we could produce motor neurons, but do we know how to make motor neurons? And that's the second problem that people solved, or developmental biologists solved over several decades. So this is a top view of actually developing chick embryo. This would be the brain inside the head, and then here you see this long tube, that's the developing spinal cord. And then if you cut cross sections through the spinal cord, so this is relatively early, so it looks like a tube that has an opening inside. You can basically see all the different cells that will give rise to different types of neurons in the spinal cord. And importantly, researchers identified several signals that are important in specifying cells to become spinal cord. Actually, one signal is a derivative of vitamin A called retinoic acid that is secreted from this region called paraxylene mesoderm and instructs the cells in the vicinity that they should become a spinal cord tissue. And then, in a sort of similar way, like in a Cartesian system where you have to specify rostrocaudal and dorsal-ventral axes, you also have to specify different cell types along the dorsal-ventral axis of the neural tube. Motor neurons are actually born very ventrally in the cells, which means like on the belly side of the spinal cord. And it has been found that there is a molecule or protein which is secreted from the ventral floor plate and notochord, which has been named sonic hedgehog. And the cells that are exposed to high concentrations of this sonic hedgehog molecule will adopt motor identity. And so what I wanted to know is whether we actually know enough about the signals that control motor neuron specification to allow me to actually recapitulate aspects of normal embryonic development in a petri dish. And so for that we took advantage of engineered mouse that expresses fluorescent protein, green fluorescent protein in all spinal motor neurons. So this is the head of the developing, this is embryo of the mouse, so you see actually it's pretty translucent and you can see the green fluorescent light here are all the cell bodies of motor neurons, and then you can see also the axons which are growing to innervate different muscles. So we took these mouse embryos, we derived embryonic stem cells